Hi everyone, in today's video lecture we are going to study about size and structure of genomes. First of all, we'll start with structure of genome. What is genome? Genome is the genetic material of an organism. So in most of the organisms, the genetic material that is present is your DNA. But in some of the organisms, for example virus, the genetic material is your RNA. The genome of the organisms are usually made up of both coding regions and the non-coding regions. Now, what is the difference between coding and non-coding regions? Coding regions are those which are going to code for amino acids, and these amino acids will be later on joined together to give us proteins. The non-coding regions are that part of the DNA which does not code for any amino acids. The term genome was given by Hans Winkler in 1920. Now, whether an organism is made up of DNA or RNA as the genetic material, they are made up of four nitrogenous bases. These are adenine, guanine, thymine, and cytosine in case of DNA, and adenine, guanine, uracil, and cytosine in case of RNA. So, in case of RNA, thymine is replaced with uracil. In most of these organisms, DNA is present as double-stranded molecule, which means it is called as duplex DNA. The double-stranded DNA molecule is usually stabilized by hydrogen bonds. In most cases, RNA is present as single-stranded, but in certain cases you can find where RNA is present in the double-stranded form. In such cases also, the double-stranded RNA molecule is stabilized by hydrogen bonds. The next important term that we have to learn is genome sequencing. Now, what is genome sequencing? It is nothing but is a complete list of sequence of nitrogenous bases present on a single DNA molecule, and that gives rise to a chromosome. And why does genome sequencing is important? The sequencing of multiple individuals is going to make us understand the genetic diversity among individuals, which means if we have sequence of these individuals, we can know what is the diversity among these individuals. First of all, we'll start with the viral genome. Now, on the right-hand side, you can see a general structure of a virus. Now, this particular virus, will have a genetic material which is covered within a protein cap. It is also called as nucleocapsid. Then they have this particular layer of protein which is called as a viral integument, and it is covered within an envelope which is called as a viral envelope. And on the surface of viral envelope, we have the viral proteins. Now, what is the function of these viral proteins? These viral proteins will help in the attachment of the virus to a particular surface so that they can enter inside the cell and they can cause infection. Now let us discuss the very important points of a viral genome. The viral genome is composed of either DNA or RNA. It means a virus can never have both DNA and RNA. Either they're going to have DNA, in such cases those are called as the DNA virus, or they can have RNA. In such cases, they are called as the RNA virus. A single virus cannot have both DNA and RNA. Either they will have DNA as the genetic material or they're going to have RNA as the genetic material. Irrespective of DNA or RNA, these genetic materials are either single-stranded or they can be double-stranded. They can be linear or they can be circular. They can be segmented or non-segmented. Now, what is the meaning of segmented and non-segmented genome? The segmented genome means when the genome has many individual unique pieces of nucleic acids. Now, you have to understand that these are not copies. These are individual pieces of nucleic acid. Now, when we have more than one copy, that means these copies are coming from one single genetic material, then these are not called as segmented DNA. Did you understand? So segmented DNA means what? It is a genome that has many individual unique pieces of nucleic acid. Like for example, influenza virus. The influenza virus has 8 single-stranded RNA molecule. So they have a segmented DNA. Did you understand? So influenza virus has a segmented genome, which means it has 8 single-stranded RNA molecule, and these are not copies of each other. These are individual unique pieces of nucleic acids. Whereas your herpes virus, it is non-segmented, they have a single DNA molecule. Now the confusion arises with HIV. Now many people, they know that HIV is basically having 2 single-stranded RNA molecule. But actually HIV genome is not considered to be segmented. Even though they have 2 single-stranded RNA molecules, they are considered to be non-segmented. Why? Because these 2 single-stranded RNA molecules are the copies of the same RNA molecule. So since they are the copies of the same RNA molecule, so they are not included under segmented genome, they are included under non-segmented genome. As I mentioned, whether it is DNA or RNA, it is enclosed within a particular protein cap, which is called as the nucleocapsid. Now the viruses which has RNA as the genetic material, as I mentioned before, these are called as RNA virus. There's a very important term that is used for such kind of virus. These are called as retroviruses. Now when it is the case of retrovirus, these viruses along with the RNA molecule which is present inside this particular protein cap are going to have a very important enzyme which is called as the reverse transcriptase enzyme. And this reverse transcriptase enzyme is going to help the RNA virus to produce its complementary DNA, which is of viral nature. Then this complementary DNA will give you viral RNA. Then this viral RNA is going to get translated into viral protein. And these are going to make up the entire viral particles and they can spread infection in the body. Next one is the prokaryotic genome. Now this is a typical prokaryotic cell, so it is basically a viral cell, you can see. So you have many, these projections outside on the surface, these are called as the pili. This is a flagellum, which is basically helping the movement of the prokaryotic cell. And then you have a capsule, or it is also called as the slime layer, which is going to protect the prokaryotic cell against unfavorable conditions. Then below that we have a cell wall. Then below that we have the plasma membrane. Inside this particular cell, mostly the genome is either circular or it is linear. Mostly it is circular genome, these prokaryotic cells they have, and sometimes it is your linear genome. And since it is a prokaryotic cell, there are no organelles that is found in the cytoplasm. Only there is one organelle that is found, and that is called as a ribosome. And we all know what is the importance of ribosome. Ribosomes are going to help in your protein translation. So when DNA gives out the mRNA, mRNA is translated in these ribosomes only. So ribosomes are also called as protein machinery. Everyone knows about it. Now, apart from this particular genome, a prokaryotic cell is going to have an extra chromosomal genetic material. In case of bacteria, it is called as the plasmid. So even the extra chromosomal genetic material, the plasmid, is going to carry very important genes. And these genes are usually expressed by the bacterial cell. Now let us discuss the important points of a prokaryotic genome. Now prokaryotes, they have usually DNA as the genetic material. Most of the prokaryotes, you can say almost all prokaryotic organisms, they have DNA as the genetic material. But in the earlier slide, we found that virus can either have DNA as the genetic material or they will have RNA as the genetic material. So if they have DNA, then those are called as the DNA virus. If they have RNA, they are called as the RNA virus or retroviruses. In archaea, they have single circular chromosome. All right. Most bacteria, as I mentioned before, also have a single circular chromosome. Most of the bacteria, but some of the bacteria, they can also have linear chromosome. Now, if in case the DNA is replicated faster and then the bacterial cell divides, then what is going to happen? A bacterial cell is going to have multiple copies of the DNA. Did you understand? So if the DNA is dividing faster at very fast rate as compared to the cell division of the bacteria, as compared to the bacterial cell division, then what is going to happen? A single bacterial cell is going to have multiple copies of the genome. Or else, if the cell is dividing faster than the DNA is dividing, then what is going to happen? The bacterial cell division is going to stop for a while so that the DNA replication is completed, so that at the end of division of the bacterial cell, the two daughter cells are going to have equal number of genetic material. And most of the prokaryotic cell, they have little repetitive DNA sequences in their genome. And as I mentioned before, apart from the DNA, apart from the chromosome, either circular or linear, these cells are going to have extra chromosomal genetic material. Now, as I mentioned, in case of your bacteria, these are called as the plasmids. And what these plasmids have, these plasmids will also carry certain genes which are going to be expressed by the prokaryotic cell. Let us discuss about the eukaryotic genome. Now look into the right side of this, we have a picture. So here, this is a particular eukaryotic cell, that is an animal cell which is not covered by a cell wall. Inside its cell, there's one very important organelle which is called as the nucleus. We have other organelles also, prokaryotic as compared to prokaryotic cells. Eukaryotic cells will have all the organelles, and one of the very important organelle is the nucleus. What do we have inside the nucleus? Inside the nucleus, we have the genetic material, which is your DNA. And this DNA is composed within a particular chromosome. And how these chromosomes are made? These chromosomes are made up of DNA, which is associated with histone proteins. So DNA along with histone proteins, they're going to, bind to each other, and they're going to, fold upon each other to give us these functional chromosomes. Now let us discuss about the eukaryotic genome. Now, eukaryotic genome, unlike the prokaryotic genome, are composed of linear DNA chromosomes, linear DNA molecules which are going to make your chromosomes. And all these chromosomes are found within the organelle which is called as the nucleus. Now, these chromosomes are made up of the DNA molecule which is found in association with your histone proteins. Then the number of the chromosome is going to be different in different species. Like for example, in human it is we have 23 pairs of chromosomes. Some other organisms are going to have either lesser than this, or they're going to have more number of chromosomes as compared to human. Now, all these organisms, they contain a multiples of a basic set of chromosomes, which is denoted as N, and that is what is called as the haploid. So all organisms are going to be made up of a set of chromosomes. Did you understand? So all organisms are made up of a basic set of chromosomes, which is denoted by N. Now, the multiples of this particular N is going to determine what is the ploidy of an organism. Say, suppose when it is said twice N, so it is called as diploid. So it is, we have two basic sets of the chromosome. So when it is two basic sets of chromosome, it becomes twice N. So what is the ploidy level of the human beings? It is your twice N. So what is N again? It is the basic set. It is the basic set of chromosome, which is denoted by the small letter N. The multiples of this basic set of chromosome is going to tell us what is the ploidy of an organism. As I have given you example, it is a twice N, so which means it is diploid. That is the ploidy level of a human being. Now, inside the human body, we have two types of cell. One is the somatic cell, another one is the germ cells. Now, the ploidy of these cells are also different. The somatic cells are diploid in nature, which means it is twice N, whereas the germ cells, those are the sperm or the egg in female. So these are basically haploid in nature. So they have one single set of chromosome. What happens during fusion of these gametes, which are haploid in nature, when they fuse together, they give us the diploid organism. So that is the definition, or that is the explanation about what is this what is that N. In addition to these chromosomes, that we have inside this particular nucleus, a eukaryotic cell, a typical eukaryotic cell will have the extra chromosomal genetic material just like a prokaryotic cell. Now, these extra chromosomal genetic material are confined within two organelles. Now, if it is an animal cell, it is confined within an organelle which is called as mitochondria. If it is a plant cell, then the extra chromosomal genetic material is confined within an organelle which is called as chloroplast. So apart from the DNA that we have inside the nucleus, we also have genetic material. These are called as extra chromosomal genetic material. And these are either present in mitochondria in case of animal cell, or it is present inside chloroplast when it is your plant cell. Unlike prokaryotes, eukaryotes have, eukaryotes have exons and introns and variable number of repetitive DNA. In mammals and plants, the majority of genome is composed of repetitive DNA. Now, let us discuss about coding sequences and non-coding sequences. Now, as I mentioned before, the coding sequences or the coding regions are that part of the DNA which usually codes for an amino acid, and these amino acids are going to join together to give us functional proteins. These coding sequences are also called as exons. Now, in the earlier slides, since I told you that unlike the prokaryotic cells, eukaryotic cells or the eukaryotic genomes are composed of exons and introns. Now, what are exons then? Exons are the coding sequences or the coding regions of the DNA molecule. Now, the proportion of the coding regions, it goes on decreasing as the complexity of an organism increases, and only 2% of the human genome that codes for protein. What are non-coding sequences? Now, since coding sequences are the exons, then the non-coding sequences are the introns. That means these introns, they do not code for any amino acids, and plus before the mRNA is translated into proteins, introns has to be removed. Otherwise, introns have to be removed. Now, these non-coding sequences are not only introns, they can be regulatory regions and your repetitive DNA also. Non-coding sequences, they almost make 98% of the human genome. Now, there are two categories of repetitive DNA in the genome. One is called as the tandem repeat, another one is called as the interspersed repeats. Let us discuss what are these. The tandem repeats. Tandem repeats are short non-coding sequences that are repeated head to tail. Let's check in the right hand side. Now there is a particular sequence which is called as CTGA. Then again it is repeated CTGA. Again it is repeated CTGA. Again there is a repetition of CTGA. You'll see there is no other nitrogenous bases present in between. So this is what is called as tandem repeat. So these are short non-coding sequences which are repeated head to tail. It is of two types. One is called as the microsatellite, another one is called as the mini satellite. What is the difference? Microsatellites will be having around 1 to 9 base pairs repeats. And what about mini satellites? They will be having 10 to 100 base pairs of repeats. Tandem repeats, they make up around 40% of the human genome. And tandem repeats, they can be functional also. Like for example, in the telomeric region, so these are the telomeric region of a particular chromosome. So telomeric region, they have a repeat, a tandem repeat which says TTAGGG. Now these tandem repeat is going to play a very important role in protecting the ends of a chromosome. Understood? So these ends of the chromosome, these are called as what? These are called as the telomeric regions. And these telomeric regions, they have this tandem repeat which says TTAGGG. And what is the function of this particular tandem repeat? It is going to protect the ends of these particular chromosome. And why this tandem repeats arises? Tandem repeats arises because of unequal crossing over and gene conversion. So you can see these are the tandem repeats. So it is, repeated head to tail. Next one is the interspersed repeats or transposomal elements. Now, what are transposomal elements? These are the sequences of DNA which can change its location in the genome. Did you understand? So that's why they are called as transposable elements, or they are also called as transposons. They have another name which is called as a jumping genes. Now, why they are called as jumping genes? Because these are the DNA sequences which can change the location in the genome. Now, how they change the location in the genome? There are two mechanisms. The first mechanism is called as copy and paste mechanism. What is this copy and paste? That means a transposable element is going to make a copy of it. Then this particular copy will go and insert themselves in another location of the chromosome. So that is called as the copy and paste mechanism. What is the second mechanism? The second mechanism is what? They get removed from one part of the chromosome, one part of the DNA. They move from that particular location and they insert themselves into a new location. So there is no formation of a copy. It is simply removal of that transposon from that particular side of the DNA. Then it gets inserted into the new location. So this is the second mechanism. So the movement or the changing of location in the genome of these transposons, that involves two mechanisms. The first mechanism is called as the copy paste mechanism. Second mechanism is called as excise and insertion. Now in this particular diagram, this is a diagrammatic representation how this transposition occurs. So you can see this is a target sequence. Okay, so this is a target sequence A, B, C, D. Then this transposable element has come and it gets inserted within this particular gene sequence. So duplicated gene target in sequence, and this is how this particular transposition occurs. Now, what is the importance of these transposomal elements? It leads to genome evolution in eukaryotes. All right, so that is the importance of transposomal elements. What is the importance again? It leads to genome evolution because number one, the insertion can disrupt a gene function. All right, so we had a gene function like we had a particular gene sequence like ATGC. You're going to insert one transposable element in between that. Now you have disrupted the gene function. So ATGC was functional not just because of the insertion of a transposable element within that particular sequence. It has become non-functional. So their insertion can disrupt gene function. What is it? It can lead to homologous recombination. What is the third one? It can also shuffle exons and regulatory sequences to new location. Like for example, a particular gene is always silent. Now because of transposition, if a particular silent gene comes in contact with the promoter region, then what is going to happen? That promoter region is going to allow the silent gene to express. Did you understand? So it can lead to shuffling of exons or regulatory sequences to new location, and that can also change the function of a particular gene. Do you understand? So transposable elements can change the location in the genome. There are two mechanisms. One is a copy paste mechanism. Second one is excise and insertion at a new location. And this transposable element is going to lead to genome evolution. And how? They can disrupt gene functions, they can lead to homologous recombination, or they can reshuffle the exons and regulatory sequences to a new location, so which is also going to change the function of a particular gene. Next topic we will discuss is the genome size. Now what is genome size? It is the total amount of DNA that is present. Did you understand? Now, this total amount of DNA which is present within a single set of chromosome. Remember, in earlier slides, I have mentioned about the small letter n. So organisms, they usually are counted as, they usually have a single set of chromosome. The multiples of this single set of chromosome is going to tell us what is the ploidy of an organism. So what is the genome size? It is the number of DNA which is present within a single set of chromosome, which is called as the haploid. Now, with certain minor exceptions, the nuclear genome remains constant within a species. Like, for example, if we take Homo sapiens, so we have 23 pairs of chromosomes. So these 23 pairs of chromosomes will remain constant in all the members of a given species, with minor exceptions. What kind of minor exceptions? Like, for example, if there are some sort of, chromosomal aberration. Now, like suppose a person can have a more than 46 chromosomes, a person can have less than 46 chromosomes, all right? So these are chromosomal aberration. So these are exceptions. So apart from these, what happens, all the individual of a given species, the amount of nuclear genome always remains the same. Now, how to measure the genome size? Genome size is either measured in terms of mass, all right? It is either measured in terms of mass or it is measured in terms of nucleotide base pairs. If in case it is measured in terms of mass, then the unit that is used is called as the picogram. So here I've given you 1 picogram is equal to 10 to the power minus 12 gram. And if in case it is measured in the number of nucleotide bases, then we're going to use 2 units. One is called as the kilobases. 1 kilobase is equal to 1000 base pairs. Or we're going to use megabases. If in that case, 1 MB is equal to 1000 KB. All right. So measurement is either done in mass, then it is picogram. Or if it is done in nucleotide base pairs, then either it is kilobases or megabases. Now here are certain statistics, like genome size of bacteriophage and virus range from few thousand bases to several hundred kilobases. Bacterial genome range from 0.5 MB to 10 MB. Eukaryotic genomes are diverse, from like 10 MB, which is present in some fungi, to more than 100,000 MB in certain plants. Human is approximately made up of 3.2 billion nucleotides. Just imagine how big is our DNA, which is divided into 24 linear molecules or chromosomes. Genome size in eukaryotes is poorly correlated with complexity of an organism. For example, here I have given you example of amoeba. You will see the size of the amoeba genome is like 70,000 MB. So you can imagine how big is the genome of an amoeba. But we all know that the genetic complexity of amoeba is not so much. It is not very complex. It is very simple. So we cannot say a higher the amount of genomic size is going to lead to higher complexity of organism. That there is no correlation, which means there is no correlation. Genome size can be big, but the complexity can be also less. Now, why is that so? Maybe it is because of the presence of huge amount of repetitive DNA in it. Did you understand? So the repetitive DNA also plays an important role in defining the complexity of an organism. So an organism can have huge amount of genome, but the complexity is very, very less as compared to many other organisms. So this is all about your structure of the genome and the size of the genome. Thank you.